Hello students, welcome back. Welcome back to my biology online classes. Today I am going to discuss with you about RNA, ribonucleic acid, another genetic material. In my previous all episodes, I have already discussed with you about DNA, and we had successfully discussed that DNA acts as a genetic material. But not only DNA acts as a genetic material, there is also another nucleic acid. DNA, the full form of DNA is deoxyribonucleic acid, and full form of RNA is ribonucleic acid. All organisms like human and many viruses have DNA as their genetic material. Example, human, Drosophila, bacteriophage, yeast, E. coli, all they have DNA as their genetic material. But some bacteria, of example, MS2, Q beta, a number of animal viruses, for instance as polioviruses, human immunodeficiency virus, HIV, and a number of plant viruses such as TMV, tobacco mosaic virus, Burley yellow dwarf virus, have RNA as their genetic material. A series of experiments proved that the genetic material contains two types of nucleic acid. One is DNA and another is RNA. Of the two, DNA is the genetic material of all living organisms, and in some viruses, RNA acts as genetic material. Fine. Now, let's concentrate about the interrelationship of DNA and RNA, the two types of nucleic acids, how they are related with each other. DNA contains all the informations needed to maintain a cell's entire processes, but these precious blueprints never leave the protected nucleus. Then how this valuable information, how all these data are transmitted to the body of the cell where they are put to use? And the answer is by the way of RNA. RNA molecules play essential role in the transfer of genetic information during protein synthesis and the control of gene expression. The diverse functions of RNA molecules in living organisms also includes the enzymatic activity of ribozymes and the storage of genetic information in RNA viruses and viroids. So, RNA may be genetic or non-genetic, catalytic or non-catalytic, and may be coding or non-coding. Now, let's concentrate about thermodynamic stability of RNA structure. The primary structure of RNA refers to the sequence of nucleotides. Primary structures of RNA, sequence of nucleotides. What's about secondary structure of RNA? Secondary structure of RNA is dominated by Watson-Crick base pairing. Watson-Crick base pairing. This fundamental interaction between bases leads to the formation of double helical structures of varying length. RNA's secondary structure is generally more stable than its tertiary structure. Secondary structure is more stable than its tertiary structure. Thus, formation of secondary structures dominates the process of RNA folding. RNA's tertiary structure forms through relatively weak interactions between preformed secondary structure elements. RNA duplexes are more stable than DNA duplexes at physiological pH. Denaturation of a double-stranded helical RNA often requires higher temperatures than those required for denaturation of a DNA molecule with a comparable sequence. Now, let's concentrate about the structure of RNA. All of you know that RNA is another type of nucleic acid. RNA is another main type of nucleic acids. Nucleic acid. RNA is made up of polymers of nucleotides. So let's draw the structure of nucleotide, and then I am going to discuss about the polymeric structure of nucleotides. It is not deoxy, so here both OH, both hydroxyl groups are present. Here carbon number 1, 2, 3, 4 and 5th carbon is here. This is ribose sugar, and here with one carbon N2 base is attached, and this bond is known as glycosidic bond. Here phosphate is attached with this 5th carbon of ribose sugar, and this bond is known as phosphodiester bond. So this is structure of a single nucleotide. This is structure of a single nucleotide, and this is also structure of unit of RNA. RNA, another important information, RNA is single stranded. It is not double stranded like DNA, and as it is single stranded, Chagall's base pair rule is not applied here. Now let's concentrate about the polynucleotide structure of RNA. Cytosine, and here CH2OH, so you can see another nucleotide is formed, and this one is 3' OH end of RNA. Let's draw another nucleotide, guanine. And this one is adenine, A. Another nucleotide is attached. Here one nucleotide, this one is attached with another nucleotide, this one is attached with another nucleotide, and let's see. this one is uracil, and this one is 5' end of polynucleotide chain of RNA, and this one is 3' end. Here 3' wedge, this is free end, so this is 3' end of polynucleotide chain of RNA. All of these are phosphodiester bonds. All of these are phosphodiester bonds. Here 5' end is free, here 3' end is free, and all of these are phosphodiester bond, phosphodiester bond, phosphodiester bond, phosphodiester bond, and here these are glycosidic bond, glycosidic bond. So this is polynucleotide structure of RNA.